Good afternoon, everyone. I'm here to talk about our new focus session for this week, which is going to be about the nephron to prepare you for the urinary system portion of our lecture. So the agenda, I'm going to talk about just the nephron, some of the anatomy of the nephron, a little bit of the physiology, kind of like the functions. I'll talk about the flow of basically our urine or our fluids becoming urine. And then I'll talk about reabsorption and secretion, and kind of what those terms mean and where things get reabsorbed or secreted in our body as they move through the urinary system, specifically the nephron. Okay, so what is a nephron? A nephron is what's considered the functional unit of the kidney. If you've ever heard the term functional unit before, essentially it is the structure in the kidney that does the function of the kidney. So we know that the kidney has multiple functions. One of the main things that it's there to do is to filter our blood, our fluids, um, to make sure that we remove everything that we don't need and then maintain things that we need. The nephron does it all. Without the nephron, there is no filtration, there is no reabsorption, no secretion out of our body and into our body. So the big parts for the nephron, broken up into two chunks, the renal corpuscle, which is this round pouch right here, and that includes the Bowman's capsule and the glomerulus. And then the renal tubule, which is basically the tubes, proximal tubule, the loop of Henle, and the distal tubule. The collecting duct is here. However, the collecting duct is not part of a nephron. There are multiple nephrons that feed into one collecting duct. The collecting duct is just there to receive what's left over after reabsorption and secretion so that it can leave the kidney, go into the ureters, and then into the urinary bladder. Filtration occurs in the renal corpuscle. So above here, you have your afferent and efferent arterioles, so blood vessels. And then the glomerulus has a lot of pores that allows things like water, sodium, drugs, amino acids, things like that to get through, but it prevents blood cells and large proteins from coming through. Once that stuff leaves, the rest of the blood is then goes out the efferent arteriole, and it's not shown here, but I'll show in a different picture. That blood vessel then loops around and keeps going through and across the tubules because some of that stuff that was removed or filtered out in the glomerulus will get reabsorbed into the blood based on what we need. Um, and then the rest of it, the renal tubule, that includes the proximal and distal tubules and the loop of Henle. And this is where all of your reabsorption and secretion occurs in the body. This is also where some of our hormones will act to retain water, retain sodium, that kind of thing. There are two types of nephrons. You have the cortical nephrons, which make up primarily 80% of all nephrons in our kidneys. And the way that I remember it the most is that these nephrons mostly stay in the renal cortex. So this nephron structure image you see here, you'll see that you have a collecting duct in the middle of both of these nephrons. However, one, you can see that the loop of Henle barely enters into the medulla, so it's very short. These primarily are used to produce urine for us. And then the juxtamedullary nephrons, that makes up about 20% of our nephrons. Here on the left, you see how this loop of Henle goes much deeper into the medulla. The reason for that is that this helps us maintain our sodium to water balance. Again, majority of our nephrons are there to produce urine, remove waste. The 20% of the nephrons, they'll still do some of these functions, but primarily they're there to maintain the water sodium balance in our body. If you get asked a question like this on the exam, I think the easiest way to remember is that the cortical nephrons are mostly just in the renal cortex, whereas the juxtamedullary cross into the medulla. So it's kind of in the word right there. Okay, so flow through our nephron. Kind of like I mentioned before, blood will come in through the afferent arteriole, and it'll then kind of run through different pores in the glomerulus, and things like water, drugs, antibiotics, um, waste, and also like electrolytes, like the sodium, potassium, that kind of thing. They will be able to be filtered out of the glomerulus, and then the remaining blood and proteins stay in the blood, and then they will then be circulated through and be involved in the reabsorption. That's this red loop right here. Um, the glomerulus is the filter. The Bowman's capsule is basically the pouch where all of that filtrate goes into, and that allows it to then start going through the tubules. The first stop of your flow is through the proximal tubule. It's proximal because it's the closest to the glomerulus. Then you also have the distal tubule, which is the furthest from the glomerulus. In between the two, you have the loop of Henle. The loop of Henle is primarily involved in water retention, and also sodium retention. And it's very interesting how this works. I'll talk about that here in the next slide. So it goes through these two loops. In between this loop for the two tubes, the distal tubule is the last stop before it hits the collecting duct. Once it hits the collecting duct, like I mentioned, that's not part of your nephron. Multiple nephrons will dump into this collecting duct, and then this will eventually leave the kidney, going to the ureters and the urinary bladder. The biggest, one of the more important parts of this chapter, this unit, is understanding uh, where things get absorbed, where things get secreted, what we keep in the body versus what we try to get rid of. Um, we later on, maybe next week on my chapter, I'll talk about the more of how things get transported across membranes. This is kind of bio like 181, 182 stuff. Um, but we have things that I can talk about later. But primarily, what we want to focus on is where in the nephron do these things get absorbed and secreted. So definitions: reabsorption means reabsorbing into the body is water and electrolytes leaving the nephron to go back into the body. Kind of mentioned that a few times. Secretion is things in our body being released and put into the nephron so that it can be combined with the urine to get rid of, through our urinary bladder. So the biggest thing that happens in the proximal tubule, about 65% of your reabsorption occurs. The reabsorption, it'll reabsorb things like amino acid, bicarbonate, water, some of your electrolytes like potassium and chlorides and sodium. But the most important thing that it can do for us is reabsorb glucose. So I kind of mentioned it down here. The body does not waste glucose. It is a evolutionary trait for our body to maintain glucose because it's a very valuable energy source. Um, Back in the day when food was scarce, so you had to go hunting or gathering, if you found a sugar source, your body wanted to maintain that uh, at any cost because we can easily pull that glucose energy and use it in our body to escape a predator, to maybe hunt or something along those lines, or just to survive. But the body will always reabsorb glucose. If you have any glucose in your urine, a lot of things have already failed in your kidneys. Usually it's not something that's just like, oh, glucose, you have glucose in the urine. Okay, we can kind of help resolve it. By this point, something has gone horribly wrong in the kidneys, um, because your body should be trying to absorb this 100% of the time. And most of this occurs in the PCT. Glucose is always reabsorbed, always. So once these things go through the proximal tubule, you'll start going into the loop of Henle. The loop of Henle is divided into I'll say two different parts, really it's kind of three. You have the descending loop, which you can kind of see here on the left side, and then the descending loop kind of goes from a very thick segment to a thin segment. And then you have the ascending loop, which starts as a thin segment, but then becomes a thicker segment. So what happens here is a process called counter current exchange, and it's in your notes. Might be an interesting topic that you may not understand right away. It took me a while to kind of get it, and I can't tell you that I 100% understand it still. But the general theory is that in the descending loop of Henle, water will get reabsorbed into our blood vessels, but there aren't any channels for sodium to go into. So you remember that water tries to follow sodium, um, and to kind of balance themselves out. In this case, remember that, in the glomerulus, we filtered out a lot of our sodium, potassium, etc., but we still have our proteins and our blood cells. Things like albumin are still in the blood vessel, and that's going to draw in the water back into the blood vessel. But the sodium will not be able to go with it. What this does is, as you remove more and more water on the descending loop, the concentration of sodium or salt increases by a large factor. Once you get to this thin layer, the sodium is now able to kind of escape the nephron and get reabsorbed into the system here. Um, and so you're very heavily concentrated in sodium here in the thin segment. But then what happens is once you get back into the ascending loop, you're so heavily concentrated here. The ascending loop actually has active transport channels for sodium. And so now all the rest of the sodium kind of gets reabsorbed back into the blood, since we are highly concentrated in water and not so concentrated in sodium. So it creates this little effect where you like highly concentrate sodium here in the loop to then force it out later. It's again the reason behind why the body does that. I'm not 100% sure. It's a little bit higher level for this class and also for my level of learning. But the main thing here is that water leaves first, sodium becomes heavily concentrated, and then once you get to these ascending loop portions, there are more active channels for the sodium to then merge and come back into the blood vessel. So you'll go, let's say concentration of 200 up here. As you go lower, you might have 600, then 900, let's say 1200. And then as the sodium leaves, you start rebalancing here your water sodium, and then you'll eventually get back to the 200, let's say milligrams of sodium that you had up here. So at the end, you still end up the same, but the process helps us to get a lot of water back into our blood vessel, and then later we get sodium to then balance out between the two loops. Um, but again, the primary function of the loop of Henle is to help with water sodium balance. Um, there's also another. I'll just keep going. So after the loop of Henle, you get into the distal tubule. Oh, another thing I didn't mention, sorry about that, was in the proximal tubule. Secretion also occurs. So some of your waste products start getting into your nephron. So things like hydrogen, ammonia, will start entering in the proximal tubule. Then when you get to the distal tubule, you'll still have, the conservation, the reabsorption of sodium, of any water that didn't get reabsorbed here in the loop of Henle, any bicarbonate, and then we'll dump out any excess, or we'll secrete any excess ammonia, urea, uric acid, hydrogen, potassium, anything that your body doesn't need, it will secrete back into the loop of Henle. There's a some there are some let's say vitamins. There's a list of vitamins where some vitamins our body will um, keep whether we need it or not, kind of like keep it now and then use it later. And there's other vitamins that if you don't need a certain vitamin, I'll just say vitamin A. I don't recall the list off the top of my head. But if I take in vitamin A and I don't need it, my body's going to immediately kick it out. It's one of those things where my body's not designed to retain vitamin A. It's designed to use it or lose it. And so if I do not use that vitamin A, it's immediately getting dumped into the nephron and getting excreted from the body. Another thing that does get secreted from the body here is things like drugs and antibiotics. So the reason that we have the urinalysis test is because drugs should not naturally be part of our body. We don't necessarily absorb them. We do dissolve them. We do use some of the chemicals in the drug to, relieve our headaches, relieve back pain, whatever. But at the end of it all, the body will get rid of the drug and drug byproducts, and because we don't retain them. And so it's urine analysis is a very easy way to determine if someone's used drugs because our body will get rid of those drugs, especially if it doesn't need them anymore. Things like ibuprofen, expires every 8 hours or so. That's getting removed from your body in 8 hours. Other drugs, I'll call them illicit drugs or illegal drugs, they do the same thing. And so once we leave the distal tubule, the last part of, at this point, we should have absorbed or reabsorbed everything that we needed. We have secreted everything that we don't need. But the collecting duct does one last okay, let me double check everything. Do I really want to get rid of this water and this sodium? If your body still thinks or still wants more water, or still needs more water, more sodium, this is where the hormones come in. So ADH, if you remember the antidiuretic hormone, it'll act on the collecting duct, the brain basically telling the nephron, or in this case, the collecting duct, I still want you to retain more water. I'm not happy with the amount of water that we got back during the normal filtration and renal tubule section, so retain more water. If for some reason the body also wants more electrolytes, it'll pull sodium and potassium also out of the collecting duct, but also in the distal tubule, or the DCT, distal convoluted tubule. That's done primarily by aldosterone. And then the other thing is parathyroid hormone helps with calcium retention. That acts both in the proximal tubule, in the loop of Henle, but primarily in the distal tubule. So if you're asked a question about hormones and where they primarily act, hormones primarily act on the collecting duct and the DCT, or the distal convoluted tubule. The reason for that is that the beginning phases, we should be able to get most of the water, sodium, electrolytes, glucose out that we need to get it reabsorbed back into the body. But let's say the brain does a double check at the very end of the cycle to make sure that, nope, I do want to keep this water, I do want to keep this sodium. Then those hormones can get released into the body to then tell the collecting ducts and the distal tubule to retain more water and more sodium. Um, and again, last thing I need to focus on, the glucose is reabsorbed at 100%. If that's not a question on the exam, well, if it is a question on the exam, ideally you get this right, because this is the only thing that we absorb 100% of the time. We absorb some of the other stuff too, but we will always try to prioritize keeping all the glucose in the body no matter what. This image here is from the Amoeba Sister YouTube video for the excretory system, which is another name for urinary system. So if you're curious about how they worked through this, I found it as a very entertaining video when I was a student, and this is arguably one of the best images I've seen of reabsorption and secretion in any other video I've ever seen. So feel free to take a look at that one. Okay, that's all I have for everybody. Thank you very much for attending the focus session. I will post the keyword key question for the hybrid students here with at the very end of the announcement. But if you have any questions, please feel free to reach out. If any suggestions or anything like that, reach out. We've only got a few weeks left in the semester, just 2 more units. We got urinary, reproductive, and then the final, and then you'll be done. Good chatting with you all. Talk to you soon.